Good day, everyone, and welcome to the STEM Lockdown Digital School. Presenting to you guys is Katlego Matibe, and I will be presenting the Grade 10 Life Sciences lesson to you today. So, guys, let us welcome our new members. And for those who are joining us from the previous or who have been with us from the previous lessons, welcome. Now, guys, remember to use my email address is at ls.wisnets@gmail.com for any questions that you might have regarding life sciences or any help. I would gladly help you. And if you want me to share slides with you, I'm willing to do so. Or if you want me to share some question papers or worksheets with you guys, I will help you guys. Now, as you know that yesterday we started off with a new topic, which is on cells and cell division. It falls under the strand life at the molecular, cellular, and tissue level. We touched on microscopy, and unfortunately we didn't get to finish, but today we are going to finish it off. So today we are going to finish off the microscopy section by doing the magnification calculations. And we're going to start off with the cells, which are the basic units of life. So that is cell structure and function. Remember to find our YouTube live streaming link. It's on the chat. So you guys can use the chat section to ask any questions that you might have or to tell me if I am not audible or if I'm moving too fast. All right, guys. So at the end of this lesson, what I will be expecting from you guys is to be able to define concepts that are highlighted in bold black, to know how to calculate magnification. That is for your practical, guys. So when you get back to school, you'll be given practical worksheets or you'll do practicals. So you'll be asked to calculate the magnifications. Or even in a test or your exams, your paper 3 will be based on your practicals. So you should know this. And I want you guys to be able to understand the structure, function, and location of the cell wall, to know the chemical composition of the cell wall, and lastly, to recognize the difference between a plant and an animal cell. Now, just to quickly recap, as I said yesterday, we started off with microscopy. We mentioned that all organisms are made up of cells, and cells are very small. Therefore, magnifying instruments such as lenses and microscopes are used to view them. Remember, we said that cells are not macroscopic, but microscopic because we cannot see them with our naked eye. And we talked about scientists who helped with the discovery of cells and discovery of or development of microscopes. We mentioned Robert Hooke, who used the light microscope to examine non-living cork cells. And this was in 1665. So he used the light microscope to examine non-living cork cells, and he said that these cork cells were in a box-like structures, and he named them cells. So he's the one who came up with the word cells. And then we talked about Antony van Leeuwenhoek, who was the first person to observe living cells using a microscope. And then the development of the cell theory was from the study of microscopic cells from other scientists that I said we shouldn't know of, but just know that they contributed to the development of the cell theory, which has a few functions that cells derive from other cells and they derive by cell division. We talked about that. And then we talked about microscopes. Now, this was a very interesting section of the topic. So we said that by using a light microscope, the simple features of cells can be studied. And the light microscope uses a beam of light focused by various glass lenses. And then we also talked about the electron microscope. Now the of magnification than the ordinary light microscope. Therefore, it allows us to see very small structures inside the cells. Now, these microscopes, the electron microscope, they use a beam of electrons focused by electromagnets to magnify objects instead of light rays and lenses. So we mentioned two types of electron microscopes, but we actually have more than two. And we said these two are the scanning electron microscope, or in abbreviation form or abbreviated form as SEM, and the transmission electron microscope, which is denoted by TEM in its abbreviated form. So the scanning electron microscope, it scans the outside of the cells. It only shows us the outside of the cells, and the transmission, it shows us the inside of the cell by allowing light to pass through that cell. And you guys asked a question yesterday and you said, do we use slides for the electron microscopes? And I said, no, we don't, but it depends on the type of microscope. So no, we don't use glass slides, but we use grids. Now, grids are small objects that look like punched paper to collect the sample that is screened. So the preparations for the electron microscope, like preparing the samples, it takes time or days even before one can view that sample under the microscope. And remember, we said that the object must be dead when using the electron microscope. So using the electron microscope, we use dead organisms, and we do not use glass slides. And for the light microscope, we talked about the parts. Now the parts of the light microscope, or the compound light microscope, we said those are, it has the optical parts which contain the lenses. That is the eyepiece or the ocular lens, the objectives. Now these are the ocular parts. They contain the lenses for you guys to, you use these parts to view or to see. And then it has the mechanical parts, which provide support and movement. And that is the stage clips, the base, the arm, the focusing knobs, and the body tube. And then we have the illuminating parts, which focuses light. That is the mirror or other light source, the diaphragm, and the condenser. Now, I can't believe I said we cannot adjust light yesterday. What a wow. Because we are talking about a light microscope. So I quickly answered that question without even thinking. And I apologize to you guys. It is a light microscope. So of course we have to be able to adjust the amount of light that goes through it. As simple as that. It is a light microscope. So you can increase the brightness or you can dim the light. It depends on your preference and how you'll be able to view the sample. Now, guys, when you're using the light microscope, you can use your phones to take a picture of the slide. Remember your phones, if you put it on flash mode and you take a picture, you can actually see the structure of the cell that you are looking at. So if in a class you are like 20 and you have 4 microscopes and your teacher asks you to draw what you see. So for drawing what you see, you can just take a picture using your phone if they do not mind. can take a picture using your phone in flash mode, and then you'll see the structure as you will see it using the microscope. So you just take a picture with your phone, and then voila, you have the structure. Now that is what we covered yesterday. Now to continue, we ended here. We are about to start with magnification. So we know that microscopes magnify an image using a lens found in the eyepiece, which is also known as the ocular lens. Now the image is further magnified by the objective lens, and we said that the objective lens can vary in sizes. May have the 5 times or the 10 times objective lens. No, actually, I meant to say the ocular lens can be the 5 times, the 10 times, or the 15 times. So Pile is asking if we will do the biomes here. No, we will not do the biomes here. You guys should have covered that before the lockdown. That is in term 1. This is term 2 stuff. So the ocular lens has the 5 times, the 10 times, or the 15 times lenses, and the objective lens has the 10 times or the 40 times and the other ones. So the magnification of a microscope, how we calculate it is by saying, or by taking the magnification power of the eyepiece and multiplying it by the power of the objective lens. So if the eyepiece magnification is 5 times and the objective lens magnification is 10 times, then the image of the object viewed under the microscope will then be 50 times bigger than the object. Megan is asking if you also calculate the microscope field of view. Yes, you'll be asked to calculate that, but it depends on your examiner if they prefer you calculating it, but you should know the calculations for that. So if you have the eyepiece magnification that is 5 times and the objective lens magnification that is 10 times, then the overall magnification will be the power of the eyepiece multiplied by the power of the objective, which is 5 multiplied by 10. It will give you 50 times the original size. So 50 is your original magnification, which will be bigger than the object, obviously. So that will be what you'll be seeing, because microscopes magnifies an image. Now we also have the field of view, like Megan was asking now. So maybe some of you were wondering what that is. Now the diameter of the field of view under low power magnification is about 3.5 meters. Before we go there, you might be asking, or you might be thinking, what is this field of view? Now the field of view is the area that is visible when you look through a microscope. So that whole area is called the field of view. When you look through a microscope, that circle is called the field of view. All right. So it is the area that is visible when you look through. Did you guys get that? But you guys have your textbooks and it is also there. So the diameter of the field of view under low power magnification is about 3.5 millimeters, but this differs. And as the magnification increases, the field of view becomes smaller. The field of view becomes smaller. It differs microscopes. So it's not an accurate number, but it is about 3.5 to 4.5 millimeters. All right. So as you increase your magnification, the field of view becomes smaller or it decreases. So you can calculate the size of a microscope field of view under low power So I'm using multiple textbooks. There are Oxford textbook, which is also free. I'm sorry, it might be because of my connectivity this side. If I'm breaking up, please listen carefully. It might be my connection. So I'm using the Siavula, which is free online, and that is awesome. We'd like to thank the Siavula group for making it available for all of us free of charge. And I'm using the Oxford and also via Africa. Those are free. So as I was saying, you can calculate the size of a microscope field of view under low power magnification by using a clear metric ruler with millimeter markings on top of the stage. So your stage has a ruler and it is in millimeters. So once the size of the field of view is known, we can estimate the size of the object being viewed under the microscope. So let's take, for instance, at 10 times magnification, the field of view is 1 millimeter. So if the magnification is increased to 100 times, what will the new field of view be? So we have So Megan is asking if we use a metric ruler. Yes, we do. Now, if we have a 10 times magnification with a field of view of 1, and then the magnification is increased to 100 times, what will the new field of view be? Now, this is the working I got from the Siavula textbook. So remember, they gave us 1 millimeter field of view at 10 times magnification. So this is low power magnification. What will the new field of view be? So for the new field of view, you are going to calculate it by saying the current magnification divided by the new magnification, all multiplied by the current field of view. So our current magnification is 10, and our current field of view is 1 millimeter. Now the new one is the 100 times magnification, because we said that it is what happened? Sorry guys, Zoom took me out. Now I don't know what's happening. Guys, I'm sorry about this. Zoom took me out. It's because of my connectivity this side. Network is not on my side. Can you guys see the slides and can you hear me? because I feel like I'm back. All right, sorry about that. Oops, technology. So as I was saying, to calculate the new field of view, we're going to say the current magnification divided by the new one, which is 10 divided by 100 multiplied by the current field of view, which is 1 millimeter. So our new field of view will be 0.1 millimeter. So remember, at the more magnification we have, the smaller or the lesser the field of view. So this is a smaller field of view at a higher magnification. So this is to show you that as magnification increases, the field of view decreases. So that is how we calculate your field of view. Your textbook might have another formula, and it says that to calculate of the specimen, you have to get the fraction of the field of view covered by the image, and then you multiply it by the diameter of the field of view. So the diameter of the field of view is what you see, and it's what you measure on the ruler. So if you have let me say the 1.8 millimeters. All right, if you have 1.8 millimeters of field of view, that is the diameter of the field of view, and then the length of the specimen in drawing, or the fraction of the field of view covered by the image will be given to you. Now, so let's say the image covers 1/5 of the diameter. So 1/5, that is a fraction. It's 1/5. So in decimal form, it will be 0.2. Then you'll say that the actual length of the specimen is the fraction of the field of view covered by the image multiplied by the diameter. So the diameter will be 1.8 millimeters, and your fraction will be 0.2, 1/5. All right. So you say 0.2 multiplied by 1.8 millimeters, and you get your actual length of the specimen. So that is how we calculate the actual length of the specimen using the field of view. I don't know if your textbooks say otherwise, but that is what I had. The magnification of the drawing, or the size of the object. Yeah? So to determine the size of the magnified specimen, you need the actual size of the object drawn and the actual size of the drawing. Now, the magnification of the drawing or the size of the specimen can be calculated using the formula size of the specimen or object equals to the drawing size divided by the object size. And the object size, that is going to be your total magnification. That is the object size. So the drawing size, that is your drawing, divided by the total magnification or the object size. So for example, if your object has an actual length of 2000 micrometers and your drawing of that object has a length of 4 centimeters, your drawing. Nah? So what you see and what you drew, you measure your own drawing. So it has a length of 4 centimeters. So you use your ruler, you get 4 centimeters. Then you can calculate the magnification of the drawing as follows. So first, the 4 centimeters must be converted into micrometers, because the actual length of the drawing or of the object is given in micrometers, and that was 2000 micrometers. So first you convert the 4 centimeters and you say 4 multiplied by 10,000 So centimeters to micrometers, you multiply by 10,000. So you say 4 centimeters multiplied by 10,000, it gives you 40,000 micrometers. And then you use the formula to calculate the size. So the size of the specimen equals to the drawing size divided by the object size, which is 40,000 micrometers divided by 2,000 micrometers, and you get the size of the specimen, which will be 20. I hope my calculations are correct. So 40,000 divided by 2,000, it gives you 20. That is how you calculate the magnification of the drawing, or the size of the magnified specimen. Now, calculating the magnification and size using a scale bar. So to determine the actual size of the object whose image or micrograph is viewed using a scale bar. So we will give you a micrograph in a test or an exam, and the micrograph will have numbers below it. So use those numbers provided to calculate your magnification and size, your actual sizes. Use that. Now we can use the following formula to calculate the actual size, which is actual size equals to the measured size on the diagram, that will be in millimeters, multiplied by the length of the scale bar, which will be in micrometers. The length of the scale bar will be provided to you guys on the diagram. So, and also the measured, oh, you divide everything by the measured length of the scale bar. So the actual size of the object, to get it, you use this formula. You say measured size of the diagram, or measured size of the object on the diagram in millimeters, multiplied by the length of the scale bar, that will be in micrometers, all divided by the measured length of the scale bar, that will be in millimeters. So this is an example. Now, so as you can see this drawing here, we want the actual size of this cell organelle. So from A to B, there's a micrograph. So what we mean by a micrograph, this is a picture of a micrograph. It was taken when scientists viewed a cell using a microscope. So this is a micrograph. And I took this particular example from the Siavula textbook also. As I told you, it is free online, so you can use it. Now, to measure or to calculate the actual size of this organelle, which is from A to B, you first measure the length AB shown in the diagram. So you first measure this length using your ruler, and it should be in millimeters. So this should be approximately 20 millimeters. You work out the length AB, given that the measured length of the scale bar is approximately 5 millimeters. So the length of the scale bar is approximately 5 millimeters. Now work out the length of AB, so the actual size of this organelle here. So the length of the scale bar you will be given. So as you can see here, you have 500 nanometers. This is the number or the length of the scale bar. So we said the length of the scale bar can be in micrometers or nanometers, like it for this case, or in this case, it is in nanometers. And it is given to you guys, so you do not have to determine it. And the measured length of the scale bar, you simply divided. So work out the length AB given that the measured length of the scale bar is approximately 5 millimeters. So work out the length AB. So we have this formula, the size or the actual size equals to the length of AB on the diagram measured divided by the length of the scale or multiplied by the number on scale bar. The number on the scale bar. So we have 20 millimeters, that is the length of AB, divided by the length that was given to us. It was said to be 5 millimeters. You divide it by 5 millimeters and you multiply by the number on the scale bar, which is 500 nanometers. This is also given on the diagram. So you guys should look at this micrograph that you'll be given. Sometimes it will be 2 micrometers or whatever the size may be. So that is the number on the scale bar. So you divide 20 millimeters by 5 millimeters and you then you convert this nanometers to micrometers. You just divide it by 1,000 and you get 2 micrometers. So from nanometers to micrometers, you divide by micrometers. Are you guys fine with all these calculations? Do you have questions? If you do not have any questions on the calculations, then we are moving on to our new topic, which is on the cells, the basic units of life. And we are going to start with the cell structure and functions. Are you guys fine with the calculations? All right. Now moving on to cells. We've been preaching that cells are made up of inorganic and organic compounds or molecules. And those molecules, examples of those are your inorganic molecules or compounds. Examples of those are your minerals. Water is also an example of that. That's what we talked about. And organic compounds, your carbohydrates, vitamins, lipids, proteins, and enzymes, and others. So cells are made up of all of those inorganic and organic compounds. And inside these cells are small structures that carry out various functions. Now, these structures are called organelles. So inside our cells, we have organelles. It is similar to humans. So our human body system has organs. So cells also have organelles. So the organization of cells is in such a way that they form complex and efficient organisms. And those organisms are, examples of those are humans, other animals, your plants. So this is due to the organization of these cells. Now the flow diagram describes this organization. We know that atoms, 2 or more atoms make molecules, and 2 or more molecules make up cells. 2 or more cells make up tissues, and 2 or more tissues working together make up organs. And 2 or more organs working together or functioning together make up a system. And 2 or more systems functioning together make an organism. And 2 or more organisms functioning together or working together then make up an ecosystem. So this is the organization of cells. We said that they are organized in such a way that they form complex and efficient organisms, which then work together to form an ecosystem. So the ecosystem is everything we see around us. So they all started off as atoms. Now we know that cells differ in sizes and shapes. So some have a regular shape and some an irregular shape. Now these shapes and sizes define the functions or some of these functions of the cells, all right? So these shapes also define some of these functions that the cells have or do. Now we have two types of cells, or we're going to talk about two types, the plant cells and the animal cells. So for the plant cells, we know that they have certain structures that are not found in animal cells. For instance, all plant cells are surrounded by a cell wall. And we know that these cells are also made up of a protoplasm, which is all the living parts of the cell. So the protoplasm is all the living parts of the cell. This protoplasm is surrounded by the cell wall, and it is made up of the cytoplasm and the nucleus. Now, all of these organelles, we are going to go into more detail or focus more on them tomorrow, which is Wednesday and Thursday. Yeah, so we are going to talk more about those or explain further their functions further. So you might be asking yourself what a cytoplasm is or what a nucleus is. We'll talk more about that. So plant cells also have plastids, and they have a single cell or single large vacuole. We have 10 minutes remaining, guys. So let us move or rush through this. So plant cells have plastids and the presence of a single large vacuole. And these two are features that make the plant cells unique, or they are unique to plant cells. So meaning that they are not found in other cells. So you cannot find plastids in an animal cell, and also an animal cell does not have a large vacuole. Now there are three types of plastids, which are the chloroplast. You are familiar with the chloroplast. We have the leucoplast and the chromoplast. Now the plant cell has a regular shape because of the presence of this cell wall. Remember we said cells have regular and an irregular shape. So a plant cell has a regular shape because of the presence of a plant cell, of a cell wall. Excuse me for that. So there's a diagram of a plant cell. On the outermost, on the outside of the plant cell, we have the cell wall. So you see it has a regular shape. A regular shape is the one that you can name, or you can tell that this is a rectangle or a square or a circle. An irregular shape, you do not know what shape it is. So it has a chloroplast. So this is part of the plastids. It has a nucleus. It has ribosomes. It has a cell membrane. And then it has the cytoplasm. We said it has the protoplasm, has the cytoplasm and the nucleus. So the cytoplasm consists of all these other organelles. It has the ER, the mitochondrion, and the large vacuole. This is the large vacuole. Now let's talk about the animal cells. So animal cells have no cell wall. Remember, the plant cells have a cell wall. Animal cells do not have a cell wall. So it has an irregular shape because of the lack of this cell wall. It also has the protoplasm, which is made up of the cytoplasm and the nucleus. So this is the, it shares, this is the similarity it has with the plant cell. They both have the protoplasm. Now the cytoplasm contains other organelles such as the cell membrane, the mitochondria, the ER, the Golgi apparatus, and the ribosomes, and many more. So it contains other organelles. And the nucleus is the same as the one in the plant cell. So the nucleus is not that different from that of the plant cell. And as I mentioned, animal cells have small vacuoles as compared to plant cells. Remember, plant cells have a single, one large vacuole. So the outer structure is the cell membrane and not the cell wall. It has an irregular shape, and it has the cytoplasm. It contains all of these organelles and the nucleus. So this is the animal cell structure. It has no cell wall, and it has an irregular shape. So guys, before I move on to the structure of the cell wall, I didn't put up a slide of your homework. So what I want you guys to do is to give me the comparison or a table denoting the differences between the animal cell and the plant cell. So tabulate the differences between the animal cell and the plant cell. Now the structure of each component. We're going to talk about the structure of each of these components or each of these organelles. We're going to start off with the plant cell wall, because the animal does not have a cell wall. So the cell wall is a rigid outer layer in plant cells. It surrounds the membrane, the cell membrane. It is non-living and is made up of both organic and inorganic compounds. It is permeable to water and mineral salts. We said that the word permeable means that it allows certain substances to pass through it. Now, the openings on the cell wall contain strands of cytoplasm called the plasmodesmata. This should be in bold black. The plasmodesmata that connect adjacent cells. Now, the cell wall is made up of three parts: the middle lamella, the primary wall, and the secondary wall. Now, this is a structure of the cell wall. It has the middle lamella, the primary cell wall, and just after the primary cell wall is the secondary wall, and then your plasma membranes and that contains your phospholipids and other compounds. So the primary cell wall is made up of cellulose. Remember, we said the cell wall is made up of both organic and inorganic compounds. So it's made up of cellulose, and cellulose is an example of carbohydrates. So cellulose is a polysaccharide, and it also has soluble proteins, which is another type of organic compound. Now, the functions of cell walls are that since it is found on the outside, the most important function is that it provides protection to the cell. It provides protection to the cell. So it provides all the contents of the cell with protection. It encloses the cell, giving the cell its shape. So it provides shape, it provides protection. It also provides a framework and support for the cell. The middle lamella binds the cells together. And lastly, the cell wall is permeable, so it is involved in transport, allowing most substances to pass through it. So these are the functions of the cell wall. Now, guys, the takeaway message is that all cells are surrounded by a cell membrane. We said that cells have different sizes, shapes, and structures in order to carry out specialized functions. The cell wall is found only in plant cells and is made up of cellulose, which is a type of carbohydrate. And the cell wall gives the plants shape, support, and protection. These are the functions of the cell wall. And the animal cell has an irregular shape due to the lack of a cell wall. So as I said, guys, your homework, tabulate the differences between animal cells and plant cells. Now, these are the references I used. So these are the references of all the things that I used, all the images, and these are the links to the textbooks. You guys have been asking about the textbooks. So these are the textbooks I used, Siavula and the Oxford textbook, and also the Via Africa. Now we will be continuing with cell structure and function. So more organelles. We'll be focusing on more organelles. So you may state 5 differences, because Megan is asking how many differences must we state. You can state 5 differences of plant cells versus animal cells. So guys, do remember to follow us on all social media platforms at Africa Teen Geeks. So that is your YouTube, your Facebook, your Instagram, Twitter. Tell your friends to also tune in and follow us. And thanks for tuning in, guys. This was your lesson, your Life Sciences Grade 10 lesson on the STEM Lockdown Digital School. See you guys tomorrow, same place, same time. And I hope you'll be more, because I see that class is increasing. So thanks for tuning in, guys. Enjoy the rest of your afternoon. Goodbye.